Hello to everyone and special greeting to Dr. Dipali ma'am. I am Sakshi Sahu, student of BSc 5th SEM. This is my mid-2 assignment of geology. I would like to thank Dr. Dipali ma'am for providing me this opportunity to show my skills. My topic is T cell receptor and their signaling. So, this slide includes the overview, what are the T cells, types, what is T cell receptor and T cell signaling. So first we should know that how the T cells are produced. So the T cells are produced from the process of hematopoiesis. From this, common lymphoid progenitor lead to form the small lymphocyte from which two cells are derived, that is T lymphocytes and B lymphocyte. Here T represent thymus and B represent the bone marrow derived. So what is T cell? T cells, also known as T lymphocytes, are one of the major components of adaptive immune system. Their role include directly killing the infected host cell, activating other immune cell, producing cytokinins and regulation of immune response. They are thymus derived and the precursor, but the production take place in bone marrow and further maturation take place in thymus. Then from there they migrate in the bloodstream in the lymphoid tissue where they can circulate. When any antigen came in the response given by the T cell receptor which further give adaptive immunoresponse. In this presentation we will learn about the T cell receptor in CD3, which is signaling protein associated with T cell receptor. Now we will learn about the types of T cells. Majorly it have three types, that is cytotoxic T cell, CD8 T cell. In this, cytotoxic T cell kill their target cell primarily by releasing cytotoxic granules into the cell to be killed. These cells recognized by the specific antigen such as fragment of viruses which is presented by MHC class 1 molecule that are present on the surface of nucleated cell. Another is T helper cell, TH, CD4 T cell. T helper cell have a wide range of effector function than CD8 T cell and can differentiate into many different subtypes such as TH1, TH2, TH70 and regulatory T cell. They become activated when they are presented with a peptide antigen by MHC class 2 molecules which are expressed on the surface of APCs. MHC class 2 molecule interact with a protein called CD4 on the T helper cells, which help to identify this type of cell. The role of CD4 T cell may include the activation other immune cell, releasing the cytotoxin and helping B cell to produce antibodies. They can help to shape activation and regulate the adaptive immunoresponse. Another type is memory T cell. Memory T cells are important because they are quickly expand to large number of effector T cell upon re-exposure of antigen and have a low threshold for activation. They provide immune system with a memory against previously encountered antigen. Memory of T cell may either be CD4 or CD8+. Here is the structure in which T helper cell is there, and this is CD4, cytotoxic T cell, CD8, T regulatory cell includes CD4, T receptors and CD25. And in this, cytotoxic T cell binds to MHC-C1 and T helper cell bind to MHC-C2. Now we should note that what is T cell receptor? T cell receptor is an abbreviated form of TCR present on the surface of the T cell. They are membrane spanning proteins. TCR interact with MHC peptide complex on the surface antigen producing cell or target cell. T cell antigen receptor are found only on the cell membrane. For this reason, T cell receptor were difficult to isolate in the labs and were not identified until 1983. T cell receptor consists of two polypeptide chain. The most common type is alpha beta and another which is less common type of gamma delta. A typical T cell may have as many as 20,000 receptor molecules on its membrane surface, either be alpha beta or gamma delta. An individual T cell generally have 10,000 to 30,000 identical copies of single TCR. There are majorly two types of TCR. One is alpha beta and another is gamma delta. In this first structure, we can see the two chain which is heterodimer called alpha beta chains. It has two variable. V stand for variable, C stand for constant. This is variable alpha, variable beta, constant alpha and constant beta. The red line show the disulfide bond between them. This is membrane. So this alone can't help for signaling because it is too small, only about of 3 amino acid. So the cytoplasmic tail of TCR is extremely short. As we can say, this is too short for transmitting the signaling, same as cable 2. In this, T cell is there and it is the bindation of two chain, but it can't be functioned as it is too small. So what take place next? See, T cell receptor and their signaling. T cell bind to the complex antigen to make a peptide located in a group of membrane bound MHC protein, where the T cell receptor make a contact with MHC peptide antigen on the surface of antigen presenting cell. The two cell membrane are brought close up to the opposition with one another. This adds the additional layer of complexity to the process T cell activation and further no parallel B cell signaling. Notwithstanding the additional complexity, the event of T cell activation still unfold according to the mixing strategy. So here we can note that there are two classes of T cell. One side is known as alpha beta and gamma delta. Now how this gamma delta come from? T cell receptors are generally associated with non-covalently bound with the complex of protein. So there are four further differentiation of protein, that is gamma, delta, epsilon and zeta. The alpha beta are heterodimer and gamma delta also heterodimer. Now this is a compact structure. This is the membrane and this is TCR. Alone it can't functions. So there is CD3 having two dimer chain, that is of gamma epsilon, delta epsilon and another of homodimer, that is zeta zeta. These all three togetherly only function for the signaling of T cell receptor. When they all combine together, so it help in the functioning of T cells. A second subset of T cell, instead of expressing the heterodimer receptor composed of different pair of protein and chain of gamma delta cell. Here the structure of gamma delta TCR and alpha beta TCR. Angle of gamma delta is 111 degree and alpha beta of 147 degree. This is V domain and this is the C domain. This ability of T cell to the breaking rule of MHC restriction may account for the evolution of slightly difference in angle between of antigen binding and constant region of T cell receptor, which is apparently seen in the X-ray crystallography analysis of T cell receptor. T cell signal reduction complex includes CD3. Signaling through TCR depend on complex protein referred collectively known as CD3. Each CDR3 dimer contained negatively. They both are negatively charged and alpha beta is positively charged. So these all are bind together with positive negative interaction between them. This is a systematic diagram of the process in which this is alpha beta TCR, this is a two heterodimer of gamma epsilon and delta epsilon, and this is of homodimer zeta zeta. By this process, the signaling takes place in the T cells. Thank you so much.